So Lab 4 is going to be all about what are called biologically important molecules, or just BIM. You'll often see it shortened to BIM. So BIMs are biologically important molecules, which is what gives most living things their characteristics. So we talk about 4 of these biological molecules. They are going to be used for things like storage and structures of all living things. So as you look at your objectives here, you'll notice the objectives themselves, there aren't that extensive, right? Pretty straightforward. We'll talk about organic molecules and all the different types of biologically important molecules we have. And then we'll talk a little bit about the tests and how to identify them, why we use controls. But what you will see here is that the keywords that are vocabulary words are quite extensive in this lab. So we're going to spend a little bit of time reviewing some of the content in this video, and we'll talk a little bit more about the actual experiments as we move into the next video. Hopefully it's only one more. We'll see how chatty I get. So the biologically important molecules are things that honestly most of you are probably familiar with. If you haven't ever looked at a nutrition label, for example, when you're looking at calories and how many calories come from carbohydrates or fats or proteins, those are biologically important molecules, right? Carbs have calories associated with them. We know there are different types of carbohydrates, right? You have simple sugars and more complex sugars. So in this lab, you're going to be looking at what are called reducing sugars, which are simple sugars. And then you're going to be looking at one more complex sugar, which is starch. Then we are also going to be looking at lipids, which are fats. Again, fats have calories associated to them, with them. As humans, we tend to talk more about what are called the triglycerides, but all fats are lipids. Then proteins, again, calories associated with these. We talk a lot about proteins when you talk about like muscle mass, for example, because there are a lot of proteins in our muscles. But proteins are actually used for hundreds of thousands, honestly, of different functions inside of our bodies and in all living things. So those 3 you've probably heard of. It's familiar to you. The 4th one here, though, is not something that has calories associated with it. So it's not really something that gets thrown around in normal nutrition chat. These are the nucleic acids. Now, nucleic acids are extremely important. They're very similar in structure to our biologically important molecules, so they fall into the category. And this includes things like your DNA, right? So obviously DNA important. It's just not something we tend to talk about when we look at food. So in this lab, you are going to be using test tubes in order to test our different biologically important molecules. So the test tubes in our lab are 10 milliliters and they are 10 centimeters tall, which means that makes it pretty easy for us to actually do the measurements, right? So you'll have 1 centimeter will equal 1 milliliter. So that will make things a little bit easier. So when you are using these test tubes, I will highly encourage you to make sure that you label them as clearly as you can, right? Where you're going to be doing 5 different experiments using 5 to 7 different test tubes for each one. You want to make sure you know what test you're doing, but also what number of test tube you're looking at. So be really careful with that. We also do reuse these test tubes because they are very reusable, create as less trash as possible. So when you are done with these experiments, we are going to ask that you clean out your test tubes, right? There's little test tube brushes and soap if you need it in order to clean out the test tubes. The other thing is that when writing on the test tubes, what we use is a wax pencil. Wax pencils work really well on glassware, but they don't come off with water because it's wax. If you don't understand that, you will soon. Right? So a dry paper towel actually takes off the wax pencil really easily, and we're going to ask that you please actually go through and make sure that your test tubes are clean at the end of lab so everyone can reuse them. So before we start jumping into what you're going to do in lab, what I want to do is review some of the material that you have talked about in lecture by this point in the semester, but just to give you an idea of what you're going to be looking at. So all of the biologically important molecules are considered organic molecules. Organic molecules are those that contain carbon-carbon bonds and or carbon-hydrogen bonds. So not every single molecule that has carbon in it is considered organic, but those with carbon-carbon bonds and the carbon-hydrogen bonds, are considered organic. So this branch of organic chemistry is a whole different ball game. You can choose to take that whole class and talk about how all of these molecules interact. Basically, what we need to talk about here is really how these things bond together, and then a little bit about the structures that they form from that. In organic chemistry, we'll talk way more about those details. So, starting with bonding. All right, again, in lecture, you've talked a little bit about bonds. We're going to look at the three major types of bonds, but the one that we're going to focus on the most here in this lab, because it's what's used to build our complex biological molecules, are called covalent bonds. So covalent bonds will form between two different atoms by using their valence electrons. So again, in lecture, you've talked a little bit about how all of the atoms have electrons that are around, kind of floating around the outside of the nucleus. Now, the ones that are farther away are going to be called the valence electrons. So when we look at these electrons being shared, it helps to complete the outer shell of this atom. So if we look at this first example here, we have an oxygen, which is the red circle, that is bound to a hydrogen, which is the little white circle. This line in the middle represents a covalent bond, meaning that these two atoms are sharing two electrons. So that oxygen will also then pick up another hydrogen to form water. Right? So water is made of two covalent bonds between oxygen and then two hydrogens. You can also see in this graphic that there is an example of a double bond. So in this case, carbon will covalently bond with oxygen, but they will actually form a double bond, which means 4 electrons will actually be shared between these two molecules. Now, based upon the electronegativity of an atom and a molecule, they don't always share the atoms don't always share electrons equally. So this results in what you can already see in this graphic between polar bonds and polar covalent bonds and non-polar covalent bonds. So polar covalent bonds are going to share the electrons not in an equal manner. So water's a great example of a polar molecule. You can see that this little notation right here, the weird little like half figure 8 with a plus, means partial. So this is a partially positive hydrogen. So both the hydrogens take on a partial positive charge, as where oxygen takes on a relatively strong partial negative charge. Now this is not as strong as ions, which we'll talk about in a second, but it is enough to make a difference in the molecule. And this polar covalent bond will make molecules hydrophilic, meaning that they do interact well with water. And that is quite frankly because water itself is a hydrophilic molecule. It's a polar molecule. So then the other example you see here is the non-polar. All right, so non-polar covalent bonds mean that the electrons are shared equally. So when you see something like methane, which is the example here, right, methane, carbon, 4 hydrogens, all the hydrogens are spread around equally. The electrons are basically distributed equally. There is no overall charge being felt on different parts of this molecule. So this makes them hydrophobic, right? So hydrophobic molecules are going to be those that are resistant to water, right? They interact well with each other, but they do not interact well with water. And it's because of the way that the charges are distributed here. Without that charge, right, there's no way to interact with water. So again, covalent bonds are going to be used to actually build all of our biological molecules. Now we also can talk about the two other types of bonds. Not as important in this lab, but a good review for you anyway, because you know, your final exam's cumulative. So the second type of bond is called ionic. So ionic bonds form between ions, right? So in this example, we're using sodium chloride because sodium chloride is literally the example in every single book I have ever used in my entire life, where sodium sends one of these electrons from its outer shell to chlorine, where chlorine picks it up, Then chlorine has those 8 electrons in its valence shell, and it is happy and complete because of that. And now sodium has an empty outer shell, so we look at the second shell, which now has 8 electrons, so it is considered happy. So what happens here is we end up with charges, right? Sodium ends up with a positive charge because it has one more proton than it does electrons, and chlorine ends up with a negative charge because it has one more electron than it does protons. And then there is an interaction between these, right? You have an interaction between the sodium and the chlorine. They hang out together. That is an ionic bond. Now, hydrogen bonds are one of the most complicated to actually write a definition for. I've been struggling with this for my entire teaching career. Hydrogen bonds are going to be an interaction between already covalently bound molecules. All right, so in this example, we're actually looking at DNA. This is one piece of our DNA, and you will see there's a hydrogen here and a hydrogen here. Those hydrogens have slightly positive charges. Now they are then going to be attracted to something that has a slightly negative charge, or so this, in this case, this nitrogen is slightly negative. It's attracted to this hydrogen. Oxygen is slightly negative. It is attracted to this positive hydrogen. That forms a hydrogen bond. So basically this is just an interaction between two separate pieces that are going to form, give a little bit of structure. So proteins will have a strong structure formed by hydrogen bonds. Our DNA is literally held together by hydrogen bonds. So hydrogen bonds are extremely important for our structure and function of our molecules. But what we're going to talk about here is really the covalent bonds, right? How do we build our biologically important molecules? So in order to build a molecule, we have to form a covalent bond. So as you talked about in lecture, as you will see in your lab book, there is a specific type of reaction that happens when we are building a covalent bond. So dehydration synthesis is a type of reaction where we dehydrate, literally, like we pull water out between two monomers and then we put them together. All right, so remember, monomers are going to be a simple building block of a polymer, and we're going to put those together to form something a little bit bigger. So the dehydration synthesis is going to occur when we take out a hydrogen from one monomer and a hydroxide or an OH group from another monomer, and that will give us water. Water is removed, and we will form covalent bonds between these two monomers. Now, in order to do this, we typically do need some energy. All right, energy is going to have to put into this, and we'll talk a little bit more about energy in our enzyme lab, actually. So then the other thing we need to do is we need to take our bonds and break them, right? So we need a hydrolysis reaction, which is kind of the reverse. We're going to take a covalent bond, we're going to snip it in half, and then we are going to put the OH and H groups back in. All right, so we're going to take water and split it. So hydrolysis, literally hydro means water, lysis means to break. We are breaking water and we are breaking our bond. By doing this, we actually release some energy, which again, we'll talk a little bit more about as we get into our enzyme lab.